So before we get into today's lecture, by the way, we're going to talk about DNA and the molecular basis of inheritance. So what I want to do before we get going is show you my tie. So I'm wearing a special necktie today, so you can see. I'll sort of show this to you, and you're probably thinking, "Oh, cool! It's a DNA tie." And anyway, I'm going to come back to this tie kind of at the end of the lecture and talk about once we have the chance to talk about DNA structure a little bit here. So hopefully you've had a good look at that. So I'll come back to that in a bit. And so today's a little bit of history, a little bit of chemistry, and a few other things in here. It's actually one of my favorite lectures. I think a lot of this stuff is really fascinating in terms of the scientific method and discovery. It's very exciting stuff. And it's a nice way to spring into the next few units where we're going to talk mostly about DNA and RNA and proteins and those kind of things. So going way back, 150 years or so, you probably know about this guy, Mendel. He's famous for his genetic studies and growing peas, apparently at least 30,000 peas. He was looking at the traits, were they yellow, were they green, were they wrinkled, were they smooth, and looking at the flowers. And he learned a whole bunch of things about genetics. And this was way before we understood anything about the chemistry. We didn't understand anything about the molecular basis. He just had some rules about inheritance and dominant and recessive alleles and those kind of things. And so very important contribution to genetics. But like I said, in terms of the molecular biology part of it, it was lacking. We just didn't have the knowledge about the molecular aspect of DNA and those kind of things. So bring forward a few years, there was this guy, Mischner. He was a Swiss doctor, and he was looking at and isolated this stuff from the nuclei of white blood cells. And so here's actually a couple of interesting things about this, is that, so it turns out white blood cells have large nuclei, so it's easy to isolate the nuclei. And so where do you get white blood cells from? It turns out we get them from pus. And so that's actually where he was getting his white blood cells from. He would actually, in the hospital, tell people, save your bandages if it has lots of pus on it for me. And I'm sure this was a very smelly job. But it worked, and he isolated this stuff. He called it nuclein, sort of in celebration of the nucleus, and found it was rich in phosphorus. So that was kind of what was known back then. And so a few more years later, we're kind of at the birth of really the modern era of chemistry. And people were able to isolate these compounds a bit more and realize that this nuclein was actually made of two things. It was made of proteins and this other substance they called nucleic acids because they were acidic. Other discoveries at the time, somebody found out that sperm cells were very rich in nuclein. So people were thinking maybe this has something to do with inheritance or genetics or something. And I don't think the word genetics was even a word back then. But the whole concept of heredity was being discussed in terms of some of these scientific enlightenments. So skip ahead a little bit forward, and the chemistry of DNA was starting to get worked out in some ways, and we'll come back to that in a minute. But there were some interesting experiments going on in various places in the world. And one of the experiments I want to tell you about is this one of Frederick Griffith. So he was looking at bacteria, Streptococcus pneumonia. So you can probably tell from the name, this causes pneumonia. And it turns out there were a couple of different strains of this. One of them they called the R strain or the rough strain. The other was the smooth. And it was called rough and smooth because that's actually what it looked like on a petri dish. The smooth one grew these sort of larger colonies, and they were smooth looking. And the rough ones were smaller, and they called them rough. And the difference is that the smooth ones have a capsule. So if you remember a capsule was a glycocalyx. And we called those sugar coats. And there were two types. We talked about capsules and slime layers. This is way back in topic 5. And it turns out that these allow the cells to stick to things, and they allow the cells to evade the host immune system. So it means that the smooth ones here, these ones here, are pathogenic, and they can kill mice and make humans sick as well, and the rough ones do not. So he was experimenting with mice, and you can see here's kind of what was known, is that these S ones with the capsule, you inject them into mice, and the mice unfortunately dies. The R ones are non-pathogenic because the immune system can mop these things up relatively easily, and you get a living, healthy mouse. So here's where the interesting parts go into this experiment. So it turns out, okay, let's see, do these cells have to be alive to kill the mouse? Yes, it turns out that if you kill the S cells by heat and you inject them into the mice die. But here's the really interesting part, is if you kill the S cells and mix them in with the R cells, the mice die. And when you look at to see what killed the mice, you get living S cells. So what he was looking at is he realized he converted R cells into S cells. And so he called this transformation. He transformed the R cells, and he believed that that transformation maybe has something to do with the hereditary material. So he called it the transforming principle. And like I said, the chemistry of what's going on at the time wasn't really fully understood. In terms of that nuclein stuff and these nucleic acids, it wasn't really known what was transforming things. So skip forward a few more years, people had been talking about genes and inheritance and Mendel, and they were like, what are these genes made of, right? So people were thinking proteins. A few people were thinking nucleic acids and DNA, but this was kind of the question mark. People were thinking proteins, and a lot of people had good money on proteins because think about proteins, they're way more complex. There are 20 different amino acids. The diversity of proteins is immense. It seems to make sense that would be a great place to have our genetic material. DNA, on the other hand, you probably know, has only 4 different nucleotides, right? We've got the letters of the genetic code, A, T, and C, and G. So people are thinking it was not a very complex structure, and maybe it was just there structurally holding the genes together or something. People didn't really know. So this brings us forward. This is almost 20 years later, and we had some scientists, I think they're in Chicago, but 2 of them actually had Canadian connections, Oswald Avery and Colin McLeod, and they decided to take that experiment and take it to another level. So what they did was they took these S cells and they used some chemistry and broke them apart. And so using different chemical techniques, they isolated the proteins, the RNA, the DNA, the carbohydrates, and the lipids. And then they're like, let's mix that with the R cells. And let's see if any of these things will transform those R cells into S cells. And so they did this. This was kind of like 5 mini experiments. And what was the answer? The answer was that DNA was the transforming principle. And therefore, this was probably the hereditary material. And so this was a good experiment. A lot of people were convinced. Not everyone was convinced. There were still people kind of debating this and saying, well, what if they were sloppy, or what if there's something we don't understand? Because how could DNA possibly be the hereditary material? Like I said, lots of healthy debate around this, but a lot of people were convinced at this point. But the question was how? How on earth does DNA have information in it? How does it code for genes? This is a really, like I said, doesn't seem like it's a very complex chemical. So one more experiment I want to show you a few years later that really was the final experiment between the end of the 40s and early 50s. People were debating it. Most people were coming around, but they just needed a little bit more evidence. So enter Hershey and Chase. This is not the chocolate bar Hershey. This is Alfred Hershey and Martha Chase, and they were collaborating and working with bacteriophage. So you remember bacteriophage, these are viruses that infect bacteria. We talked about them already, and there they are. And they kind of look like the lunar landers. And remember the structure of this thing. We've got a little bit of DNA in here, and then the rest of that is made out of protein. So now we have something that is conveying genetic material because it's going to make new viruses, and it's made only of 2 things. It's made only of proteins, and it's made only of DNA. So what do we do? This is 1952, and well, it turns out we can use radioactive nucleotides. So this ended up being 2 experiments, and I'll show you what they are, and they're actually very clever. So the first experiment, they were using radioactive sulfur, sulfur 35. So it turns out sulfur is found only in proteins. It's not found in DNA, not found in nucleic acids. It's found in 2 of the amino acids. So what you do is you grow up some viruses in radioactive sulfur, right? And so they're going to incorporate that into their proteins. Okay, so that's part 1. Part 2 is you take those viruses and let them infect some new E. coli. So there we go. You can see E. coli here is being infected. So this is E. coli or E. coli getting infected by viruses. And then you shake it up. So I think they used a blender. And what you can do is centrifuge this. And so if you centrifuge around 15,000 RPM, you're gonna pellet heavy things. And by heavy things, I mean cells. Anything lighter than cells, like viruses, are not going to get pelleted. So the question is, what made it into the cell? Was it the DNA or was it the protein? So what you can do, you centrifuge this. You can see the pellet is going to have the bacterial cells and anything else heavy. And the liquid part, that's called the supernatant, is going to have what didn't make it into the cells. So it turns out you can separate these things and you can show that the liquid remained radioactive and the bacterial cells were not. So this shows that the protein, at least, did not go inside the cell. And probably protein was not the genetic material. But you got to follow this up with a second experiment. This experiment, like I said, it shows nothing. It shows that the protein is not the genetic material. So we want to do the second experiment to confirm that it is, in fact, DNA. So experiment 2, we're using radioactive phosphorus 32. I've used this stuff before. It's pretty radioactive stuff. And there we go. We've got E. coli getting infected again. But this time, our viruses have radioactive DNA and not radioactive proteins. So same experiment, you infect E. coli, you shake it up, centrifuge it, and now the radioactive material is in the pellet. So the DNA is in the pellet. So here's kind of the summary of that experiment. The conclusion was that since the DNA is the thing that's getting inside the host cell, the DNA must contain instructions, and therefore the DNA must be the genetic material. So yay for DNA, except for, like I said, how does it work? People had no idea. There were lots of mysteries around DNA in the early 1950s, and they were all about to get cracked, and really, really interesting stories. So by the way, let's just talk a little bit about the chemistry of DNA. And we've talked a little bit about it already way back in topic 2. So DNA is made out of nucleic acids, and nucleic acids are made of basically 3 things. Phosphate groups. You can see this is a phosphate. It's basically a phosphorus atom surrounded by some oxygens. We have a sugar. So sugar, it's a pentose sugar, meaning it has 5 carbons. So here's our pentose sugar. You can see there's a little note here that says this is a deoxy sugar, and that's part of DNA instead of RNA. We'll talk about the differences between DNA and RNA in a later lecture. And we have these nitrogenous bases. So the 4 letters of the genetic code. So you can take this, this whole thing here as a nucleotide. You put those 3 things together, and we put a bunch of nucleotides together. And here is one nucleotide right there. And you have basically DNA. And so you can see there's the 4 different nitrogenous bases, thymine, guanine, cytosine, and adenine. And so we're going to talk a little bit about how we figured out what is going on here. So this is kind of what was understood of the chemistry at the time. We didn't understand anything whether it was double-stranded or if there were 4 strands or how information was conveyed in there. There was no understanding at the time in 1950. But like I said, we were about to crack this. Chemists had cracked this part of the puzzle, but kind of the biological part and sort of how things are put together at a larger level was really the mystery. We just knew we had long strands of these in the cells. So a little bit more information for you on the nitrogenous bases. So they are called nitrogenous bases because they have lots of nitrogen in them. And they're bases because they actually have these amine groups. You can see these amine groups. These are actually weak bases. So good name for them. The ones with one ring are called pyrimidines, and the ones with two rings are called purines. I always get them mixed up because I think in my brain, I'm thinking the longer word should be the larger structure, but it's the other way around. The purines have a smaller word and a double ring structure. And that's what they look like. And there's other nitrogenous bases out there, by the way. We'll talk about uracil when we get to RNA. The chemical caffeine is a nitrogenous base as well. And so there's a few out there in biology, but these are the main ones of DNA. So just a little bit more terminology for you. You may have heard of people, sometimes they say nucleotide, and then once in a while people say nucleoside. You can see the difference. A nucleoside is just a sugar in a base. So this one here is a nucleoside, and the second one is a nucleoside. If you also have the phosphate, and like the third one, you're a nucleotide. So there's a whole bunch of different of these, right? If you think about, we've got RNA and DNA, we've got all those different nitrogenous bases. We can have a phosphate or no phosphate. So there's actually a lot of possibilities in terms of how we can arrange these in some ways. So it's a little bit more complex than people were thinking. You can have, and we already know that with ATP, we can have 3 phosphates, so 1, 2, 3, or we can have 2, or we can have 1. And so these molecules, these nucleic acids, they're used in 2 main areas of the cell. They're used in information and information propagation, so DNA and RNA, and they're also used in energy, so ATP, GTP, CTP, UTP, and so on. So there's our nucleic acids. We can have, like I said, you can have 1, 2, or 3 phosphates. So there's our nucleotides. We can have nucleosides. We can also have the deoxy versions, so the DNA versions. And usually what we do when we talk about them is they're abbreviated. So for example, we might talk about DATP. That would be the deoxy ATP. That would be a DNA nucleotide used for DNA synthesis. We have the normal ATP. And so that would be an RNA nucleotide, or also could be used for energy. It's a dual functional molecule as well. Sometimes when we talk about these things, we might just use the 1-letter code. So anyway, I'm just trying to go through the terminology with you. I'm not expecting you to like memorize this table or anything like that, but you should know a little bit about how the terminology works, because we are going to talk about these here, these the last ones anyway, the DNTPs, and we're going to talk about them next week a little bit. So 1950-ish, actually 1949, this guy here, Erwin Chargaff, I think he was an American, I really can't remember, but he was looking very carefully at DNA and the chemistry of DNA in a variety of organisms. And he came up with some observations that were eventually called Chargaff's rules. And everybody was fascinated by these things because they're like, is he onto something? Like, what is all this stuff, right? So he realized that if you look at 2 different organisms, their DNA was different, right? Different amounts of A, G, T, and C, right? But, so number 1 isn't as important as number 2. Number 2, he realized that the number of A's, so the amount of adenine, was equivalent to the number of thymine, and the amount of cytosine was equivalent to the amount of guanine. So what does this mean, right? Like if you had 15% adenine, you'd have 15% thymine, and then that would leave what's left over, 60%, so 30 and 30 of C and G. Nobody knew what this meant, but they call it Chargaff's rules, and everyone was talking about it, at least everybody who was interested in DNA, of which was like a lot of scientists at the time, because everyone knew whoever could crack this, the person who could crack this, we're talking about that everyone knew this would be a great biological discovery. So there's Chargaff's rules. I guess I drew them on the other screen. And like I said, the question was, what does this mean? So this is in the back of the mind of many people. So 1950s still, One of the players who ended up in this story is a guy named Maurice Wilkins. He was an X-ray crystallographer. He was working on a variety of things. Eventually, in the 1950s, he started working on DNA, and he was hiring this woman here, Rosalind Franklin. She becomes a very famous historical figure for a whole bunch of reasons, of which I'll get into in a minute. And she was very, very talented X-ray crystallographer, and I'll talk about what X-ray crystallography was in a minute. She had done her PhD, I believe, in France, and working with these X-rays, and she was probably one of the best X-ray crystallographers in the world at the time. And he didn't actually hire her directly. I think his boss hired her. And so there was a little bit of a mix-up. He thought that she was going to be like his assistant, and she was told that she was going to be, have her own lab. They kind of got off to a bad start. And then a lot of people historically have written about this. She was 1950s, a very strong, independent woman. She had to fight very hard to get to where she was in the 1950s. To give you an idea how bad some of the sexism was at the time, there were separate lunchrooms for the men and the women at the time. There were very few women who had PhDs and were into research at the time. And these were some of the things that she had to deal with. Unfortunately, the historical record shows she was probably not a very nice person. She was a bit of a jerk herself. So anyway, they're off to a bad start, but working on doing X-ray crystallography, a whole bunch of things, including DNA. So let's talk about X-ray crystallography. So X-ray crystallography uses X-rays. And what you do is you have a crystal of a sample. So this was being used for a whole bunch of different chemistry at the time. And what you do is you crystallize a sample, you hit it with these X-rays, and you do it in a certain way. And it's really quite complex. I'm not going to go into all the details. In fact, many of them I don't really fully understand myself. But if you took high school physics, you might remember that light will diffract when it hits certain sizes of slits and things like that. So what's happening is the X-rays are interacting with the atoms in the crystal, and they diffract and they produce a pattern. So this was something that people were able to do and start to understand the molecular structure of things. Now, mostly at the time, people were looking at small molecules, not something immensely complex like DNA. But Rosalind Franklin produced this image here. And this is a very, very famous historical image. And I'm going to come back to that in a minute. And I'm not equipped to fully understand exactly how you interpret this. But I'll tell you a little bit about what Watson and Crick did when they interpreted it in a minute here. So Rosalind Franklin made this beautiful image. She didn't really know what it meant. She was clueless, quite honestly. That seems to be the historical account. Some people are trying to say she almost had the answer. I don't believe that's the case from what I've read. But enter Watson and Crick. So these two guys were in London, and they were, where were they at? Cambridge or Oxford? And they were working on different projects. One of them was working on, I think, bacteriophage, and the other guy was Crick was a chemist and a very bright guy, and he had the dullest of projects you could imagine. He was studying the viscosity of water. So he had a physics background, and he was bored to tears. And anyway, him and Watson kind of had this mutual passion and curiosity. And in particular, they were very interested in DNA. And they would just talk about DNA all the time. And they even shared an office, right? So, what do you do when you share an office? You talk about things you're interested in. Both of them are interested in DNA. And so they kind of dabbled, they read things, and they went to conferences, and they heard Rosalind Franklin talk about the structure of DNA, and they asked her a few questions, and they were, at the time, they didn't know much about DNA, and they actually, she told them basically they were idiots and they didn't know anything, and they later admitted to themselves that it was true, they didn't know much about it, but that didn't stop them from starting to learn about DNA and start to look into what kind of research had been done. And 1953, they cracked it. And they did not do a single experiment themselves. and they used a whole bunch of data from other people. And I'll show you how this all came together. So one thing, which was sort of one of the later parts of the puzzle, but really important, at that point, they had learned so much about DNA, about the chemistry and the structure. And one of them went over to King's College to talk to Maurice Wilkins, and Maurice Wilkins, Rosalind Franklin had a new job lined up, and they were he said, hey, you should check this out. Franklin took this cool image. I don't know what it means. So he showed it to Watson without Franklin's permission. Some people argue that that was dishonest, but technically Franklin was working under Wilkins in his lab. So it was his data as well to share. But anyway, like I said, different people talk about how to interpret this differently. But there's some key information in this. And like I said, in terms of the crystallography, I don't quite understand all how you calculate this. But one important thing was the width of the DNA. So that was super important. How fat is DNA? Like I said, we don't know. Is it 2 strands, 7 strands, 3 strands? I don't know. So this told us that it was likely 2 strands, the DNA was. So that's hugely important because there were people working on like 7 strands and 5 strands and 3 strands and lots of weird things people were trying to sort out. It also told them it was a helical structure, which kind of just made everything fit together so much nicely in the end. And the other thing, it told them that the phosphates were on the outside. And you can see this thick band part on the outside here. And phosphates are a heavier atomic structure. And so it kind of makes sense that they would light up a little bit better on the outside of the structure. So these were 3 things, right? And you can imagine that, okay, they're like, Watson was like, wait a second, this is key information. This is huge. And like I said, Franklin did not know how to interpret this, this structure because she thought it was a useless image. Probably because she had a physics background and didn't understand a little bit about biology. But I could tell you a lot more about that if you ask me sometime. So they went and they started doing some modeling. So if you're in organic chemistry, if you take it someday, you know that you can get these model kits. And they built this massive model and they started looking at it and they realized, okay, if we had 2 purines, that's too wide. We know the dimensions now. Okay, we can't do that. What if we do a purine plus, or pyrimidine plus a pyrimidine? Because initially what they were trying to do is they were trying to, they would try to pair A plus A and G with G and so on. And they realized that, no, we need to pair a purine plus a pyrimidine. And that is perfect. No one had thought of this before. And there's a little bit around the chemistry of this whole thing in terms of people didn't quite have the chemistry of the nitrogenous bases correct. And Watson and Crick, there was another person, I'm not mentioning the story, that kind of corrected them and said, I think most people have it wrong in terms of how the forms of these nitrogenous bases are. And so they used what we now know are the correct forms. And they realized immediately, wow, look at this. A and T form perfect hydrogen bonds together. And G and C form 3 perfect hydrogen bonds together. This is incredible. And that's a purine and a pyrimidine. Perfect width, perfect chemistry. And of course, they couldn't help not thinking about Chargaff's rules. And they're like, wait a second, remember Chargaff's rules? And A is always the same amount as T, and G is always the same amount as C. So they're like, okay, we're onto something. We got it. We just have to build this thing. So they built their model, and that last piece I told you, remember, was helical. The model didn't quite fit together right until they gave it a little bit of a twist. And they built this model, and it was like beautiful. It was perfect. They knew they had it right. They put together a manuscript. Watson and Wilkins were good friends, and they sent him an advance copy because, of course, they had used some of him and Franklin's data, and Franklin and him had been working on this for years. And, you can see that quote there, where Wilkins just said, I think you're a couple of old rogues, but you may have something. As one rat to another, good racing. He knew they had it, and he thought it was brilliant. And in the end, they ended up publishing with Wilkins in the same manuscript, Nature magazine. And well, anyway, here's another possibility of how it may have happened. You can see this is saying, they're in the bar discussing the structure of DNA. And he says, I'll have a double helix, little cartoon, or maybe it happened in their office like this. Here's Watson. He says, I say, old chap, I suspect DNA may be a double helix. And Crick says, "Good show, Watson. I think we've got it." Actually, Watson was an American, so he would have an American accent. But anyway, you get the idea. So they published, Crick's wife drew this image of DNA. It was actually a 2-page paper, which is super short, published in Nature in 1953. And the scientific world was blown away. It just was so elegant, so perfect. People were almost convinced overnight. There's always some doubters. But as the data started pouring in from all sorts of, other people doing X-ray crystallography and other experiments, it was confirmed within months. Other people were confirmed 100%. If you go to Cambridge University today, you can see there's a stained glass window that was modeled after that original sketch in honor of this seminal paper. And over the next few months, Watson and Crick published a few more things that confirmed to sort of flesh out this model for everybody else. So this is I'm only scratching the surface of this story. It's really fascinating, and I'll get back to the story in a minute here. But just to sum up what is going on here with the double helix, some chemistry for you. So you can see that there's a couple of things I haven't mentioned yet. So as I mentioned here, we've got these nucleotides, right? And they join together, and you have 2 strands. So the second strand is going to pair, and so you're going to pair your A with your T and your C with your G because of the complementary hydrogen bonding. So we call the 2 strands complementary. Complementary. And the other thing that you may have noticed, and we're going to talk about this a little bit more in the next unit, is that notice I'm talking about this here having a 5' end and this one having a 3' end. And I'm going to talk about that a bit more in the next unit. But if you take a look at this, this strand here is in one orientation, and this strand here is basically upside down compared to the other strand. And in terms of the chemistry, like I said, I'll explain that 5' and 3' again in better detail next day. I'll draw you a picture. But it is that's kind of the way it is, right? One strand is backwards compared to the other. So the term that we use for this is anti-parallel. And this is something that Watson and Crick learned from doing the modeling. They realized it didn't work the one way. Okay, we'll just flip it upside down, and the second strand is anti-parallel or backwards or opposite. The last thing I think I mentioned before as well is that they felt it didn't fit together quite right until they added a bit of a twist to it. And so it famously became known as the double helix. So if you want to learn more about this, I really highly encourage you to check out Watson's book. Watson wrote this book a few years after the events, and it's a really great read. It's not that long. It's like 120 pages. It's one of the best nonfiction books I've ever read. It's got a little bit of everything. It's got science, so that's exciting. It's got drama, gossip. Watson, some people really hate Watson for writing this book because he was a little bit gossipy, and it was clear from writing the book what his thoughts were on Rosalind Franklin. But like I said, they both dished it out. She was certainly not nice to him as well. And so some people feel like he really was very sexist and rude towards her. And in some ways that's the case, but it's a very interesting personal account. And it's a good book that kind of, you get caught up in the excitement at the time, and you get into the mind of what it was like to be at least a male scientist in the 1950s. There's been many books written later on Rosalind Franklin's part of the story. She never got a chance, unfortunately, to tell her part of the story. She died, I believe it was by the end of the 1950s due to cancer, probably from all the x-rays. She was, she did not believe that x-rays would give her cancer, and she never wore protection against it. And she was exposed to them for many years in her career, and unfortunately it led to her death. So really sad, but I encourage you to check out this book. It's really, really good book, really enjoyable. And like I said, you gotta just put aside and realize, yeah, there are some antiquated ideas in there around the genders and whatnot, but I don't think I'm surprised. It was written in the 1950s. You don't have time for the book. You can rent the movie. Actually, I haven't seen it, it's a made for TV movie, but Jeff Goldblum, I kind of like him. Anyway, if you ever see the movie, let me know. You can always read a little bit about Crick's version of events. Crick actually only wrote one chapter of this in his book, and this was written many years later. And it talks about Crick had quite the career in genetics and other things for many years. So only one chapter is devoted to the discovery of DNA. So what I want to do now, just for a moment, is I'm going to go back to my tie. So take a look. Here's my tie, and you can take a close look at this tie. So I was telling you, you might be excited. I've got a DNA tie, and now you know that this is not DNA, right? So take a good look at that. We've got there's no double helix on there. It's just a swirly ball tie. I bought this years ago. I was on eBay and I saw a DNA tie, $7, and within seconds buy it now. And a few weeks in the mail it came, and I'm looking at it and I'm like, this is not DNA. So unfortunately the joke was on me. Thankfully it was only a $7 joke. And I'm trained as a biochemist. I've looked at this many times. This is no known molecule. It's just a bunch of balls and sticks put together by an artist with a swirly thing around it. And they sell it as a DNA tie, and they rip off people, unfortunately, like me. So if you're ever somewhere and you see somebody with this tie, just laugh to yourself and know that it's not a DNA tie. Anyway, so now in my case, it cost me $7, and I have a story to tell every time I give this lecture, which is kind of fun, I guess. Okay, so a few other things I want to talk about here. So one of the things that Watson and Crick realized immediately, and they wrote one sentence in that paper and then followed it up about a month later with what they thought was going on, is they said if you look at the structure of DNA, this immediately suggests a possible copying mechanism, right? In terms of, if you look at this, they understood very quickly right away that these base pairs, these A, T's, G's, and C's, could code for something. And one key thing about having genetic material is you have to copy it, right? That's pretty obvious. If you're going to copy that genetic material and give your genes to your children, and they're going to give genes to their children, there's got to be some mechanism for that information to be transmitted. And so I want to just talk a little bit about DNA replication, which is really the next unit, but a little bit about the history of it. So they realized that all you had to do if you want to make more DNA is separate the two strands. And then if you have one strand, you can easily figure out what's complementary. So if you have an A, you're going to have a T. If you have a T, you're going to have an A. If you have a C, you're going to have a G, and so on. So they're like, hey, we think we know how DNA is copied. And this wasn't proved experimentally yet. And that's kind of the last part of today's lecture, is I want to show you how this was shown experimentally by another group of scientists. So at this point, structure of DNA is out there. People are like, yep, that's got to be it. How's it copied? Watson and Crick had proposed that model, and that model actually is the middle one here, the semi-conservative model. But some people said maybe it's a conservative model. And what do we mean by that? So if you take a look, here's our parent DNA. Oopsies. Okay, let me just go back. So there's our parent DNA, and you can see it's kind of dark. And they said, well, maybe the parent DNA, it just stays together, and then you get some new DNA, and it's all new molecules. And then you can see this one replicates again, and the one on the left replicates again. Watson and Crick were postulating a semi-conservative model, meaning that each strand joins a new daughter strand, right? And so on and so on. Some people said, well, what if it's even like something different? They called it the dispersive model, where different bits and pieces of nucleotides and we're all mixed up. And I don't think anybody ever believed in the dispersive model, but it was a hypothesis, right? So what I want to do is talk about what is going on here is another experiment by a couple of Americans, Michelson and Stahl. Okay, so what I'm going to do is switch to the whiteboard here and talk a little bit about their experiment and draw you an image. But the first thing I'll let you know about their experiment is that they were growing bacteria, and they were growing them in two different types of nitrogen, nitrogen 14 and nitrogen 15. So nitrogen 15 it has another neutron. It's an isotope. It's a little bit heavier. So a little bit, that can be measured by scientists. So let me just, I'm going to switch now to the whiteboard. You may want a piece of paper. Let me just pull that up here and I'll share that in a second. I just got to open up the whiteboard and get this ready. Hold on a second here. Sorry about that. What happened to my whiteboard? Okay, there it is. And I just need to share it now. Okay, I don't know why these things sort of disappear on me, but there we go. Okay, so here's my whiteboard. We're going to call this the Messelson-Stahl experiment. So what I'm going to do is I'm going to draw some DNA, and let's see here. I'll draw two colors. Let's see here. I'll draw one of them in black and the other in blue. So they started off and they had some DNA, well, actually some bacteria, because the DNA is being replicated in bacteria. And so what they did is they replicated it in, they grew it in nitrogen 15 media. So when you have all the DNA had nitrogen 15, right? So we'll call this one heavy because it has the nitrogen 15 in it. And then what they did is they switched it to nitrogen 14 media. So nitrogen 14, I'm going to do in blue. So we know now that DNA, when it replicates, it basically separates out into two strands. And new daughter strands, new daughter DNA molecules come in and they match up and with the complementary nucleotides. So our new strands here, so hold on, I just need to make a little bit of space there at the bottom for my labels. So there's my new strands. Uh-oh, that was not the right color. I meant it to be blue. There we go, there's blue. So the new strands are a hybrid of the nitrogen 15 DNA and a hybrid of the nitrogen 14 DNA. So I'm going to call these ones medium in terms of mass or density. So I kept this in nitrogen 14 media for one more round of replication. So I'm going to just shrink this a bit here, make sure I can fit it all on the screen. So take a look, there's our original strands, and our original strands are going to separate. So we get our nitrogen 15. I'm just looking at the one on the left, and nitrogen 14, and then nitrogen 15 strand. And another nitrogen 14 strand. And of course, all the new DNA is nitrogen 14. So I'm going to draw that in. So we got nitrogen 14 and nitrogen 14, nitrogen 14, and one more. Just shrink that a tiny bit more. Nitrogen 14. Okay. So and now we have this one's medium, and this one's medium. And these new ones are lightest. So light, we'll call them, because they're all nitrogen 14. So take a look at this. We have 2 rounds of replication. And we go from heavy over here to some medium density DNA to medium plus light. Okay, so that's the whole basis of their experiment. And I'll show you, I'm going to go back to the PowerPoint now and show you what was going on with their experiment and how they measured this. So back to the PowerPoint here. Just get this up. And so there's their experiment. They're growing it in nitrogen 15, and then they grow it in nitrogen 14. And they follow 2 rounds of replication. And what they're going to do is they're going to isolate the DNA, and they are going to separate the DNA by this really expensive, high-speed type of centrifugation. So this is a centrifuge. And what they do is they use something called the cesium chloride gradient. And in that cesium chloride gradient, you can separate things by density. This thing is going really fast. We're talking about 100,000 times the G-force. And you can get DNA separated by different bands of density in this thing. So what you can do is you can do this and separate it out and see what's going on. So here's what they found. They grew the bacteria, and they found that after round 1, they had 1 medium-sized band, and after round 2, they had 2 bands, a medium-sized band and a light band. So remember, sketch I just made. After round 1, we should have 1 type of DNA. After round 2, we should have 2 types of DNA, right? And that's what they found. Let's just go back and think about those other models for a second, and you can take a look at this in the textbook. It's explained really well in the textbook, I think. If you think about these different models, right? The conservative model, the parent DNA is always together. So you should always have 2 bands. You should have the parent DNA and the daughter DNA, and that's it. You shouldn't have a 1 band and then 2 band system. Semi-conservative model, you should have 1 band for the first replication and 2 bands for the second one. So this is, this is looking good. The dispersive model, your final DNA is all going to be basically mixed, and it's going to be kind of all a medium weight. There's not going to be multiple bands. So this was, like I said, a relatively clever experiment that basically confirmed this. Now, there's been all sorts of other experiments that have confirmed this over the years. But this was basically to show that Watson and Crick were right. They guessed it, and good job. And it was just a matter for the rest of the scientific community to basically confirm a lot of what they had speculated in their model. So next topic, we are going to look at DNA replication. And so DNA replication and its basic function, you separate those 2 strands of DNA and you bring in the complementary base pairs. And you can see that's what's going on in the video here. And so complementary, remember we have A pairs with T, and G is going to pair with C. So we're going to talk about that next week. We're going to talk about all the enzymes involved. It turns out it's relatively complex. We have 6 enzymes and proteins that are involved in the DNA replication process. And and this is an important part of the course. So let me just take a look at the next slide for a second. No, the next slide was just the end. So before I let you go today, I'm just going to give you, tell you a little bit about the final exam. I am going to be giving you lots of hints and lots of promises about the final exam. And I'm hoping by the time we get to the final exam, there will be no surprises. So I encourage you to, from this lecture and on, keep a piece of paper beside you. And when I give you hints and tips about the final exam, you write those things down. There's a lot of these things you will see on the final exam. So for example, here is your first tip. This last part of the course, we're going to be talking about 3 processes: replication, transcription, and translation. So we have a lecture on each of them, and then all the other lectures are related to replication, transcription, and translation. So one of your long answer questions on the final exam will be getting you to describe one of those processes. So basically, I'm telling you, one of the questions will be on replication, transcription, or translation. And in fact, there'll be tons of questions on all those things. One of them might be a matching, one of them might be fill in the blanks. So it'll be definitely each of those processes, there will be, are worth at least 10 to 15 marks on the final exam each. So final exams are worth 80 marks. So I just told you that's about half of the exam is to understanding these processes very easily. So learn them well over the next few minutes. I'll be giving you some sample questions as we go along and talk about that as well. So one more comment, somebody's asking, is the final exam cumulative? And the answer is sort of. The final exam focuses on the last part of the course. So maybe 70%. So, and the other 30% is from the first part of the course. So what I do when I make the final exam, it's similar to your midterms. Maybe it's about double what your midterm looks like. And so it's about 40 multiple choice and 80 marks. And so out of those 40 multiple choice, I basically go through every unit and take 2 multiple choice from every single unit. And most of the written part of the exam is going to be on this last part of the course. So topics 13 all the way to topics 19. So we'll clarify that a bit more as we get closer to the final exam, but you do want to mostly focus on this last part of the course. So if there are no other questions, I'm going to sign off now, and we'll see you on Monday. Like I said, we'll talk about grades, we'll talk about midterm 2 on Monday, and a few other things. Have a great weekend.